Hello everyone, and welcome to another recorded lecture of Anatomy and Physiology 1 online. Today we will discuss Chapter 2, which is on the chemistry of life. And Chapter 2 is a bit of a review of a lot of concepts you've learned in either biology or in chemistry before. And let's look back at the first chapter when we mentioned that the body's organized, right? All living things are organized, and especially as we talk about anatomy, we'll talk a lot about how things are organized and this hierarchy of organization. And before we talk about cells or tissues or organs or organ systems, let's start at the chemical level. All right, let's start at the molecular level and let's talk about chemistry. Right, so the chemical level is the smallest or lowest level of organization that we can talk about, and chemicals are not living. Right. Chemistry is the study of matter and the interactions of matter. And anything that has mass and occupies space is matter. So there are actually 4 different states of matter. You can have a solid, a liquid, a gas, a plasma. A human body is made up of chemical matter. So the anatomy, the structure of the human body is made out of chemical matter. And the physiology or the functioning of the human body results from thousands of different chemical reactions. And these chemical reactions all have to occur in thousands of cells, or actually not thousands of cells. We could really say if your whole body, right, has you could estimate over 30 trillion cells in your body. So within a certain cell type, there have to be thousands of different chemical reactions functioning just perfectly. And the sum of all of those chemical reactions in a cell or in an organism is called metabolism. Right? Metabolism is how we use energy and how we store energy in chemical reactions. There's 2 ways we can use our energy. We could use our energy to build things up. That's called anabolism. So for example, we need energy as we build muscle. Like when you're lifting weights, for example, you have to put in energy so you can build muscle. So building muscle is anabolic. You're growing. Catabolism is breaking things down. So during digestion, we have to break down the things that we eat. That's catabolic. So the sum of anabolism and catabolism equals metabolism. So you can ask yourself, if you have a very fast metabolism, does that mean you do more catabolism or anabolism? If you have a fast metabolism, that means you break down things more than you build things. All right, so again, it's kind of a balance between anabolism and catabolism. Life is very fragile, as we know. So life as we know it depends upon the normal functioning of millions of these different kinds of chemical reactions in trillions of cells at all times of the day. So let's talk about chemistry at the most basic level. We said chemistry is the study of matter, and all matter is made up of atoms. So an atom is the smallest bit of matter we'll discuss in this class. There are 92 naturally occurring elements on Earth. For example, carbon is an element, oxygen is an element, hydrogen is an element, gold is an element. And an element is something that is a pure substance. It can't be broken down into anything more basic, right? So we can say that an element is like a pure substance that's naturally found on Earth. There are 92 naturally occurring elements, and an element is divided into atoms. An atom is the smallest piece of an element. And if you wanted to know all the elements that we have on Earth, you would look at the periodic table of elements. For example, hydrogen is an element, and it tells you the chemical symbol, it tells you the chemical name, it shows you in this picture a little diagram of what one atom looks like, and then also has the atomic number and the atomic mass, as we'll see soon. But the periodic table is very important because it organizes all the atoms of all the elements in a very organized fashion, very, very highly specific. So for example, and I'll talk about this later, but all the elements in this column over here have very common features, whereas all of these in this last column have common features. all of the elements go from smaller to larger as you go from left to right. So a lot of the properties were taken into consideration when organizing the periodic table of elements. So despite having 92 elements that are naturally found on Earth, only a few of those elements are involved in living organisms. So we can mostly, right, the major elements of the human body include oxygen, carbon, hydrogen, nitrogen. Those are the biggest. You could kind of remember CHNOPS. That's how. So for carbon, calcium, hydrogen, nitrogen, oxygen, phosphorus, and sulfur, CHNOPS, is how I always remembered it. I could kind of write that down. These are the major elements in living organisms. So carbon, calcium, hydrogen, nitrogen, oxygen, phosphorus, sulfur. Again, you don't really have to know these lesser elements, but you should know that our body does require some trace elements, like magnesium and iron, in order for us to function. So we need some trace elements, but we also have these are called the bulk elements, because these compose more than 98% of our body weight, these elements. So we can draw atoms in different ways, but the way they're usually drawn is by showing the nucleus, which is the center of every atom, and the electrons surrounding the nucleus. So there are three types of subatomic particles found in atoms. Protons have a positive charge, neutrons are neutral, so no charge, and electrons have a negative charge. And the protons and neutrons reside in the nucleus. So the positively charged protons and the neutral neutrons are in the nucleus. And then you have electrons that orbit in different energy shells. And again, this is a review from chemistry or biology. But what defines each atom, right? Atoms of different elements have different numbers of protons. And depending on the number of protons an element has, that will determine the number of electrons. And atoms, like I mentioned, of different elements have very different chemical properties. And that's how the periodic table is organized, taking those different properties into account. So this is how we draw an atom. Again, protons and neutrons in the middle, electrons orbit in different energy shells. Electrons are much lighter than protons or neutrons. Their mass is basically negligible. They don't really weigh much at all. And atoms in general are very small to see, right? So we could say that since this is really not drawn to scale, because if this little dot were protons and neutrons, electrons would be orbiting really way out here, and all of this is actually empty space. So it's interesting how most atoms are actually empty space. But yet we draw them in a simplistic way so we can see the protons and neutrons in the nucleus. So carbon, for example, has 6 protons. So we draw the 6 protons, and it has 6 neutrons, and it has 6 electrons. If it has 6 protons, that means it has 6 electrons, because all the elements are neutral. Right? So an element is neutral. So we'll soon see how to draw different arrangements of electrons more specifically, but you should remember that the first shell has 2 electrons, and every other shell can hold 8. So this carbon has 4 electrons on its outermost shell, right? 6 total, so 2 go in the middle, 1, 2, and then 4, so a total of 6. This is how we would draw a carbon atom. A hydrogen atom is just 1 proton and 1 electron. Right, that's an easy one to draw. So these are some important terms. The atomic number refers to the number of protons. So the atomic number for carbon is 6 because it has 6 protons in the nucleus. And the atomic symbol is just the letter or 2 letters that represents the element. So C is for carbon, Na is for sodium, Au is for gold. So some of them are a little more tricky. And the atomic mass is another number you should be familiar with, and that's the sum of the number of protons and neutrons in each atom. Sometimes it's not a whole number because it actually takes into account different forms of elements called isotopes, and it averages out all the different atomic masses. But in general, you can round the atomic mass to the whole number. So you could say it's 12. It's called the mass number. And that means that every nucleus of a carbon atom has a mass of 12. So if there are 6 protons, that means there must be 6 neutrons. Right? So the atomic mass is the total weight of the nucleus. It's the sum of the number of protons and neutrons. So to find out the number of neutrons, you can just take the atomic mass and you subtract the atomic number from that. And that gives you the number of neutrons in each atom. An isotope. You can have an isotope of an element if you have the same number of protons but a different number of neutrons. So for example, hydrogen is hydrogen because it has 1 proton. All right, so this is the average hydrogen atom, has 1 proton and 1 electron. But you can have an isotope of hydrogen called deuterium that actually has a neutron in it. So it has a different weight, it has a different atomic mass. So this has, whereas normally hydrogen has an atomic mass of 1, deuterium has an atomic mass of 2 because it has a proton and a neutron. Tritium is another isotope of hydrogen that has 2 neutrons. So it has an atomic mass of 3. But again, it's still hydrogen because hydrogen by default always has 1 proton. Some isotopes release energy as they decay, and those are called radioisotopes. So a radioisotope is a radioactive molecule that decays, it breaks down, and as it breaks down, it releases energy. We can use that energy in many ways. We can use it to kill cells that are growing too fast. Like for a brain tumor, we can use high beams of radioactive energy that can actually kill cells specifically with that energy. We can also use low levels of radiation for imaging, like in an MRI. Can use a radioactive dye that you inject, and you can see how the tissues are metabolizing that dye based on using this radioactivity. So atoms make up molecules. A molecule is a substance formed by 2 or more atoms bonded together by covalent bonds. A molecule can have atoms that are different or identical. For example, oxygen and hydrogen gas, as we know them, are both just pure. So oxygen gas is not just one oxygen atom. It's actually molecular oxygen, meaning it's oxygen that's bound or double bonded to another oxygen. Hydrogen gas, hydrogen never just comes alone as one H, right? It's a molecule of hydrogen, H2. A compound is a substance formed by atoms of 2 or more elements. So that means a compound is a molecule too, but it has at least 2 different kinds of elements. So if water, for example, is a compound because it has hydrogen and oxygen in it. But hydrogen gas is just a molecule because it just has 1 element of hydrogen. Right? All compounds are molecules. Not all molecules are compounds. Right? So here are some other compounds. Salt, NaCl, sodium chloride, and glucose, C6H12O6. A chemical formula expresses the chemical composition of a molecule. So there's 2 ways we can express a chemical formula. We can express the molecular formula that tells you how many of each type of atom are there in each molecule. So butane, for example, is an organic molecule that has 4 carbons and 10 hydrogens. Right? 4 carbon atoms and 10 hydrogen atoms in every butane molecule. So that's the molecular formula. The structural formula shows the linear arrangement of atoms and shows how they're connected to each other. And it's possible that you can have the same molecular formula, the same number of atoms, but they're arranged differently. So the structural formula gives you a little more information. So butane and isobutane have the same molecular formula, but a different structural formula. And we call these isomers. An isomer is something that are structures that are different, but have the same molecular formula. So if you have two molecules with the same molecular formula, but different structures, we call those isomers. So we said molecules are atoms connected by chemical bonds. But what is a chemical bond? A chemical bond is a force of attraction between two atoms that allows them to join and form a molecule or compound. And that force of attraction, right, the way that they form chemical bonds is by transferring or sharing electrons. So those negatively charged electrons that orbit around the nucleus of every atom can be transferred or shared, and that can create a chemical bond. Generally speaking, atoms are most stable with a full set of 8 electrons on their outer shell, and that is called the octet rule. Octet means 8. So all atoms, for the most part, like to have 8 outer electrons. And they try to do whatever they can to get the outer shell to have 8 electrons. The exception is with hydrogen and helium. They want to have 2 electrons on the outside because they only have 1 and 2 electrons respectively. But there are 4 different kinds of chemical bonds we'll talk about in this class. And again, chemical bonds are happening because atoms want to have a full set of electrons on their outermost shell to be most stable. So chemical bonds happen because atoms want to be more stable. They can do an ionic bond. That means electrons could be transferred. There could be electrons being shared between 2 atoms in a covalent bond. You can have a hydrogen bond, which is a very a weak attraction between sticky ends of neighboring molecules. Or you can have what are called van der Waals forces, which are very, very weak interactions that we're not going to talk too much about in this class. So ionic bonds will be the first type of bond we'll discuss. And to start, let's define an ion. So an ion is an electrically charged particle. So an ion is another way to think about an atom that has either gained or lost an electron. The best way to illustrate ionic bonds and ions is with an example of sodium chloride. So here we have an atom of sodium. It has 11 protons and 12 neutrons in its nucleus. Since it has 11 protons, we know it has to have 11 electrons. And we start by putting 2 in the first shell. The second shell has 8 electrons. And of course, we're left with 1 remaining electron on the outermost shell. Let's look at chlorine. A chlorine atom has 17 protons and 18 neutrons in its nucleus. If it has 17 protons, that means it has 17 electrons. So we can do 1, 2. The second shell has 8. And then we'll put 7 on the outermost shell. Now, what did we say about the octet rule? We said atoms are most stable, they're most happy when they have an outer shell of 8 electrons. So what happens over here is sodium says, hey chlorine, I know that you want 1 electron really badly, and it just so happens that I have an extra electron. So sodium is very willing to get rid of the electron, so chlorine can then have a stable shell, and so can sodium. Because sodium lost a negative charge, it becomes a positively charged ion. Right? So without an extra electron in its outermost shell, we call this a sodium ion with only 10 electrons. And then since chlorine gains that extra electron, right? This was a new electron just added, it now has 18 electrons and it's negatively charged. And what do we know about positive and negative? Right? They're drawn to each other. So an ionic bond is the attraction between 2 ions of opposing charges. Right? So we have 2 ions of opposite charges. You have a positive ion and a negative ion. They're attracted to each other by an ionic bond. An ionic compound, right, is a compound that is formed by ionic bonds between the ions. So 1 crystal of sodium chloride salt, right, is an arrangement of sodium ions and chloride ions joined by ionic bonds. And these ionic bonds are very, very, very strong. However, they can be broken in water. So once you dissolve salt in water, all those ions break apart and become sodium, positively charged sodium and negatively charged chloride. So that's ionic bonds. The next category are covalent bonds. So in a covalent bond, we have atoms sharing electrons. Right? Ionic was transferring, right? One gained and one lost. But in covalent bonds, we have sharing electrons. So for example, we said hydrogen wants to have 2 electrons on its outermost shell. So each hydrogen atom on its own is pretty unhappy. But when they come together and they share a pair of electrons, each of them will have 2 electrons, right? This hydrogen atom has 2, this one has 2. And we can draw a pair of shared electrons as a line. So when you draw H, line H, that means there are 2 hydrogen atoms sharing a pair of electrons. So basically what I'm saying is a flat line is a covalent bond. It's possible to also have a double covalent bond if 2 electron pairs are shared. And you see this a lot between carbon atoms, or between carbon and oxygen, or between carbon and nitrogen. So for example, carbon dioxide, you have a carbon molecule that's doubly bound, so double bonded to 2 separate oxygens. So oxygen and carbon, this oxygen and carbon are sharing 2 pairs of electrons. This oxygen therefore has 8, 1, 2, 3, 4, 5, 6, 7, 8 electrons. This carbon over here is going to have 8, 1, 2, 3, 4, 5, 6, 7, 8. Great. And this oxygen over here will have 8. 1, 2, 3, 4, 5, 6, 7, 8. So this entire molecule, all 3 atoms in this molecule is very happy because every atom has a full octet. You can also have triple bonds and share 3 pairs of electrons. So covalent bonds are bonds where electrons are being shared, but they can be shared evenly between 2 atoms or unevenly between 2 atoms. So if electrons are being shared equally, we call that a nonpolar covalent bond. So electrons are equally attracted to both nuclei. So there's an even distribution of electrons, right? It's a nice even distribution. That means nonpolar. This molecule is nonpolar since there's an even, equal distribution of electrons. However, there could be electrons that are shared unequally. For example, in a bond between oxygen and hydrogen, the electrons spend a lot more time around the oxygen than the hydrogen. Again, electrons are being shared, so this is still a bond, right? We still draw this as a flat line, and this is, again, not charged. But since electrons spend a lot more time around the oxygen, this has a partial negative character. So we draw this little script delta negative sign to show that this end of the molecule is slightly partially negative, whereas this hydrogen end is slightly partially positive. And that's because oxygen is more electronegative than hydrogen. And what electronegativity is the greediness or the desire for electrons. So this oxygen really wants to hold on to electrons very badly. So electrons spend more time on the left, so to speak, which makes this partially negative. And since there's a partial negative and a partial positive, clearly electrons are not being shared evenly. So that's what explains this polar covalent bond. Polar is unequal sharing, nonpolar is equal sharing. So nonpolar covalent bonds are very common between identical atoms or between carbon and hydrogen. So methane, for example, you have 1 carbon atom that has 4 vacancies. You have 4 hydrogen atoms, each with 1 vacancy. They come together to form 1 methane molecule. And carbon and hydrogen are not really greedy for electrons, so electrons are shared equally. Therefore, we can draw 4 covalent bonds. Each of those lines I drew were covalent bonds. And we could say that each covalent bond is nonpolar because neither carbon nor hydrogen cares more for electrons. If a molecule has nonpolar bonds, meaning electrons are shared equally, we say the entire molecule is nonpolar. And most nonpolar molecules will not dissolve easily in water. We call nonpolar molecules hydrophobic. They fear water. This is very important, right? So a nonpolar covalent bond means electrons are shared equally, and when that happens, it does not dissolve easily in water. So they're hydrophobic. Examples of nonpolar molecules are oils and fats and waxes. They don't dissolve well in water. A polar covalent bond is when you have unequal sharing of electrons, and therefore you're going to have a polar molecule. And a polar molecule usually happens when one atom, usually an oxygen or a nitrogen, hogs all the electrons away, where that one end becomes partially negative compared to the other end. So it kind of makes it like there's one end that's sticky, that each end has sticky ends. I'll explain, that will make a little more sense in a bit what the stickiness means. But molecules that have polar bonds are hydrophilic, right? Polar molecules and ions dissolve very easily in water. They're water-loving. And that can be explained by the next type of bond we'll talk about, hydrogen bonds. So this will explain that stickiness that I'm explaining in polar molecules. Hydrogen bonds are weak bonds between sticky, partially charged ends of polar molecules. So remember that water is a polar molecule. Oxygen is going to pull on the electrons a lot more than the hydrogens. That makes the oxygen partially negative. And of course, what that's going to do is it'll attract the partially positive hydrogen of a neighboring water molecule. So you have this partially negative oxygen. With this dashed line, it's making a weak hydrogen bond with this partially positive hydrogen. That's a hydrogen bond. So they occur between a partially positive hydrogen atom and a partially negative atom that's usually an oxygen or a nitrogen. And that's what we denote this dashed line as a hydrogen bond. So one of them is very weak, but one water molecule can make many hydrogen bonds. And these hydrogen bonds form and reform millions of times per second. So the hydrogen bonds that water forms with other molecules, other polar hydrophilic molecules, is essential for life. So pause here and try to answer this question. The answer is B, uneven sharing of electrons, and nonpolar molecules are usually hydrophobic. So a chemical reaction is nothing more than the formation or breaking of bonds. So a chemical reaction occurs when bonds between atoms form or break. In a reaction, we start with the reactants, the starting materials, and then we show an arrow, and then we draw the products, the ending materials. There are three basic types of chemical reactions. If you want to synthesize something, you want to build something that's anabolic, you need to build new bonds. Right? In a decomposition reaction, if you want to break down things, like catabolic reactions, you have to break bonds. Or you could also have an exchange reaction where bonds are rearranged between the atoms. Regardless, chemical reactions use or release energy. And energy is the capacity to move something, whether it's moving a muscle or moving an electron, right? Anabolic reactions require an input of energy. And that makes sense, right? If you want to build something up, if you want to build muscle, you need to put in energy. So in order to build, let's say, a protein out of its smaller building blocks, the smaller amino acids, you need to input energy, right? To attach all of these things into a bigger part. So synthesis reactions are anabolic. They require input of energy. The word to describe requiring an input of energy is called endergonic. So anabolic reactions, like synthesis reactions to build things, are endergonic. They require energy. Catabolic reactions usually release energy. So like a decomposition reaction, if I'm taking something larger and breaking it down into smaller parts, I'm going to get energy out of it. And those reactions are called exergonic. Exergonic reactions release energy. So organic compounds, organic chemistry deals with molecules that have carbon and hydrogen. Usually they have oxygen as well. And we'll talk about 4 different organic molecules in living things. They're called macromolecules because they're large. And they're also called biomolecules because they're found in all living things. So there are 4 biomolecules that are organic and macromolecules. So again, it's a lot of redundancy. But there are 4 categories you've probably learned about before. There are carbohydrates, lipids, proteins, and nucleic acids. And you should know a lot about these already from biology. So I'm going to go through them very briefly in this presentation. Inorganic compounds, in comparison, do not contain both carbon and hydrogen. So salts and water, electrolytes and minerals, all that stuff, those are inorganic compounds. So let's talk about water before we talk about all of those organic compounds, because water composes about 2/3 of our body weight, and it's a reactant and a product in many chemical reactions. Water is also really important as a lubricant in our joints. So our bones don't rub against each other because of this synovial fluid that's mostly made of water. Water is also really important for thermal stability. It helps us stabilize the internal temperature of the body, right? And that's because water has a high heat capacity. The amount of heat required to raise the water temperature is a lot. So heat doesn't change very drastically. And that has a lot to do with hydrogen bonding. Hydrogen bonds keep water intact and stable. They're not going to break apart and boil very quickly, and they're not going to widen and freeze. Water is the solvent for most solutes in the body. So a solution consists of particles called a solute that are mixed with a more abundant substance called the solvent. So again, a solution, like a sugar solution, sugar is the solute dissolved in water as the solvent. So water is needed to transport solutes throughout the body, like salts and sugars, right? They easily dissolve in water. What word describes salts and sugar and that they dissolve easily in water? What can we say about salts and sugar? They are hydrophilic, right? Because they dissolve easily in water, because they can make hydrogen bonds, right? They're polar and hydrophilic. Where is water found? Right? So we have water inside of our cells in what's called the intracellular fluid. So our cells are mostly water themselves. And also outside of the cells, we have extracellular fluid. So outside of our cells in tissues, we have water. And that could be in plasma, blood plasma, and also interstitial fluid, which is fluid between the cells in various tissues. Finally, a separate organ altogether, lymph. It's a modified version of plasma that are found between our lymph vessels. And we have a whole system, a whole network of lymphatic vessels in our body. So we have a lot of water. Mixtures of substances in water may be a solution, like I mentioned, which is a dissolved solute and a solvent. You could also have what's called a colloid. So, for example, albumin in blood plasma is an example of a colloid. Because a colloid, they're small proteins that are equally distributed in plasma. So, in water, like plasma is an example. So our plasma is like a water, but it has evenly distributed proteins like albumin. Milk is also a colloid, right? It's mostly water, but it has evenly distributed proteins like casein. A suspension is different. A suspension is you have really large particles suspended in water. So the blood cells in our blood plasma exemplify a suspension. And that's when particles are larger than 100 nanometers in size. And you can actually, it's possible for them to settle to the bottom of the tube just with the force of gravity. So especially if you use a centrifuge, you can spin down all the blood cells in a suspension to separate the cells from the plasma. Electrolytes are another important aspect of chemistry. So electrolytes are ions, which are charged particles that are easily dissolved in water. So we have a lot of these inorganic electrolytes that are crucial for our physiology. So for example, bicarbonate ions help us maintain the right acid and base in our blood. We have calcium ions that are important for muscle contraction. Phosphate ions are a major component of bones and teeth. So we have a need for electrolytes for many functions in the body. So electrolytes can be salts and minerals or acids and bases. So let's talk about acids and bases. Water, H2O, is made up of two ions, so to speak. It can be broken down into an H+, which is, in other words, just a proton, and an OH-, which is a hydroxide. So normally, most water is H2O. But 1 per million water molecules is an H+, but breaks up into an H+ and an OH-. So that's just something to note. So an acid is any proton donor. Any molecule that releases a proton in water is an acid. So an acid increases the hydrogen ion concentration. A base does the opposite, right? A base is a proton acceptor. So, for example, anything that can absorb H+ is a base. Or anything that increases the concentration of OH- is also considered a base. A base is also known as an alkaline. And acidity is expressed in terms of pH. So molarity is a way to express concentration. So pH is a measure of the hydrogen ion concentration or molarity of a solution. So a pH scale measures the acid-base balance of a solution. And the lower the pH, the lower the number, the more H+ are present. So it's a little tricky. And the more acidic. So the lower the pH, so like a pH of 2, has a lot of H+, which means it's the most acidic. Now that's because P is the negative log of, so it's an inverse relationship. The lower the pH, the more acidic and the more H+ ions there are. So the pH scale will indicate the relative acidity or alkalinity of a solution. And it ranges from 0 to 14. And anything that is between 0 and 7 is an acidic solution. pH 7 is neutral, meaning there's an equal breakdown of H+ and OH-. Anything greater than 7 is basic. Is a basic solution. Right? Anything less than 7 is acidic. The pH scale is logarithmic. So what that means is that each unit of pH is a factor of 10. So think about how much more acidic is pH 5 than pH 7. Right? So pH 5 is 10 times more acidic than pH 6, and pH 6 is 10 times more acidic than pH 7. So each unit is a factor of 10. So pH 5 is actually 100 times more acidic than pH 7. Right? So make sure that that makes sense to you. So this is a nice image that shows the whole range of pH scale. So where a 7 is neutral, you have an equal concentration of in blue is OH-, and red is H+. And again, we said 1 in every 10 million water molecules is going to have this dissociation of H+ and OH-. So pH 7 is pure water. How would it expect? It's just the 1 per 10 million molecules dissociated. If you have more H+ dissociating, right? You have more H+, then we're going to get more acidic and acidic. So hydrochloric acid is a very strong acid. Stomach acid has like a pH of 0.5. It has a lot of H pluses. It's very, very acidic. Then we have like lemon juice and beer, vinegar, tomatoes, coffee, urine, milk. And then again, pure water and tears are neutral. Human blood is slightly alkaline. It's about 7.35 to 7.45. Then you have baking soda, and then cleaning products tend to be very alkaline or basic. So try to pause here and answer this question. So the answer to this is, it's an acid, right? It's less than 7. So a buffer is a chemical solution that can stabilize pH. So a buffer can resist any large change in pH. And that's very important to maintain homeostasis, right? It requires our blood pH, for example, or our pH of all of our fluids to be a very specific number. We can't deviate from a certain normal range. So if our blood gets anywhere more alkaline at 7.45, our other parts of the body have to compensate in order to try to bring our pH back down. If our pH gets too low, that's acidosis. So our body tries to, again, compensate. We can breathe out a lot of CO2 to try to get out that acidic carbon dioxide. And you'll talk more about that in AP2, actually. But a buffer is something that can act as an acid or a base. So when the pH is high, then what do we want? Do we want an acid or a base? If the pH is too high, right, then we want an acid. We want to give up more H pluses. So the bicarbonate buffer system, right, it can act as an acid when the pH is too high, or it can act like a base when the pH is too low. So that's why buffers are really important. A salt is an ionic compound that dissociates in water but doesn't produce H plus or OH minus like an acid or a base. So like HCl is an acid because it increases H plus concentration. Potassium hydroxide is a base because it increases the OH concentration. But KCl is a salt because it just dissociates into a potassium ion and a chloride ion, right? That's what we said about salt. And those examples are joined by ionic bonds. And salts provide ions that are essential for normal body functioning, like blood clotting, also for nerve conduction. So we'll talk a lot about sodium, potassium, and calcium in this class when we talk about muscles and nerves. So organic compounds, like I mentioned, are called biomolecules in living organisms that include carbohydrates, lipids, proteins, and nucleic acids. And carbohydrates, proteins, and nucleic acids are built as long chains with repeating subunits. Lipids are a little different, but what we call these long chains are polymers. So a polymer is a long chain of monomers, and a monomer is the repeating subunit. So for example, in a pearl necklace, the pearl necklace is a polymer and each pearl is a monomer. And cells are constantly building up and breaking down these molecules. There are two really important reactions that you need to understand. Dehydration synthesis removes water to join molecules. Hydrolysis adds a water to break apart molecules. So automatically you should think one of these is going to be anabolic and one of these is going to be catabolic. Think about which is which. So a dehydration reaction happens between two monomers that need to be connected. So we're going to remove, there just happens to be hydroxyl groups at the ends of monomers. And when you remove an H2O, right, you're dehydrating it. You're removing H2O and then putting a bond in its place. So when you're putting bonds, when you're building two things and connecting them together, that's dehydration. That's anabolic. You're building things up. Right? So a synthesis reaction would utilize right, it's called synthesis, dehydration synthesis. If you want to build something, you need to do dehydration. The opposite, if you want to break something down, you're going to use water to break a bond, right? Lysis is splitting or breaking. So using water, hydro, to break the bond into two separate monomers, and that's catabolic. So this is going to be what we'll talk about for the rest of the lecture. And again, I'm going to go rather fast since you have read it, you've read it in the book already, and you've probably taken this recently. Carbohydrates contain carbon, oxygen, and hydrogen. in a 1:2 to 1 ratio. It's one carbon to every two hydrogens to one oxygen. Monosaccharides, like these guys over here, are single sugars or simple sugars. Mono means they only have one ring, a single ring sugar. These are used for short-term energy. So glucose in our blood is used for short-term energy, like minutes to hours. Glucose has a chemical formula of C6H12O6. That's our blood sugar. Pentose sugars, like deoxyribose and ribose, look like a pentagon, and they are components of DNA and RNA. Disaccharides are double sugars, di for two, and that's two simple sugars joined together. White table sugar, for example, is sucrose. It's when one glucose and one fructose attach to each other by dehydration synthesis. Then we get this covalent bond called a glycosidic bond between these two monosaccharides, and that gives us the whole molecule of sucrose. Lactose is a disaccharide that's built of a glucose attached to a galactose. So galactose and glucose, this is lactose. Maltose is two glucoses. If you are lactose intolerant, that means you cannot break down this bond. You can't break the bond between galactose and glucose. And that makes it indigestible in your system. So the lactose found in milk is indigestible for certain people, and therefore they might have to take a supplement that has the enzyme lactase. And the enzyme lactase can easily break this bond in lactose. In general, things with the suffix ose, OSE, are sugars. Finally, we have polysaccharides that are long chains of simple sugars. Poly is for many. And in animals, the most popular polysaccharide you need to know is glycogen. So glycogen is a bunch of glucoses attached together. So glycogen is a chain of a bunch of glucoses attached together by glycosidic bonds. This is how our muscles and liver store glucose. In plants, they don't have glycogen, they have starch. So polysaccharides in plants are starch, and that's again chains of glucose. And that's for long-term energy storage. Lipids are a diverse group of fatty, non-polar molecules that have carbon, hydrogen, and oxygen. They have more hydrogens and less oxygens than a carbohydrate. But both carbs and lipids have carbon, hydrogen, and oxygen in them. Lipids are for long-term energy, and they have more energy in their chemical bonds. And all lipids are hydrophobic, meaning they fear water, they do not dissolve, and all lipids are non-polar. The most abundant in our body are triglycerides. We have a lot of triglycerides in our fats. Triglycerides also make up oils in plants. We also have phospholipids in our cell membranes, and steroids. So steroids, fats, and phospholipids are all lipids. Fats and oils are made of triglycerides, and triglycerides themselves are molecules that have three fatty acids. That's what's called triglycerides. It has three fatty acids attached to an alcohol molecule of glycerol. So a triglyceride is a glycerol with three fatty acids, and these are the building blocks of fats. Fatty acids are nonpolar molecules, right? They're all hydrophobic. And like I mentioned, since they have so much energy stored in these fatty acid bonds, all these bonds between the carbons and hydrogens, they can be used for long-term energy storage. So we have special cells, adipocytes, that store all these triglycerides, all these fat droplets, in case we need it for long-term energy. Fats are also used for insulation in animals. Right? It's found in adipose tissue around the internal organs, under the skin, and inside the bone marrow. So fats are solid at room temperature if they are saturated. So saturated fats are when you have fatty acid chains where carbons are all attached to two hydrogens each. So every carbon has two hydrogens attached to it in the fatty acid chain. And because of that, that makes the fatty acid chain very straight and rigid. The straight fatty acids can then be packed together very tightly because they're so straight and rigid, and that means that saturated fats can be solid at room temperature. Conversely, in an unsaturated fat, at least one carbon in the hydrocarbon chain is bound to just one hydrogen. It's not saturated with hydrogens, right? It's unsaturated. And that means there must be a double bond. And if there's a double bond in the hydrocarbon chain, that means it's a crooked fatty acid, right? Like a little kink in the chain. And that means it can't pack together as tightly. Right? These can't pack together as tightly. So as a result, unsaturated fats are liquid at room temperature. So a steroid is a lipid with 17 of its carbon atoms arranged in 4 rings. So cholesterol is a steroid from which all other steroids are synthesized. So the others include cortisol and bile acids, which aren't shown here, but also testosterone, and estrogen and progesterone. So cholesterol is the base steroid. It's also a major component of cell membranes. And from cholesterol, we can get all the vital hormones we need. Eicosanoids are interesting. Those are a type of signaling molecule that are made from a certain type of fatty acid. And again, these are used for communication and also have roles in inflammation and blood clotting. You could have a synthetic steroid that is a variant of testosterone that athletes use to bulk up muscle quickly, but these pose major health risks. So pause here and see if you can answer this question. And the answer is A, fats. So proteins. Proteins are large molecules that are found in all living things. And proteins actually come from the Greek word proteos, which means of first importance. So proteins are definitely the most important. They are the building blocks of all living things. They're basically like the hardware. They do the things a cell needs to do. All proteins found in living organisms are made from the same 20 chemical building blocks called amino acids. So a protein is a polymer of amino acids. There are major types of proteins. They can be structural, like collagen and keratin. They can be enzymes that help catalyze chemical reactions. They can transport molecules, like hemoglobin. They can pull muscles, like myosin, or they can be protective, like antibodies. And like I said, all proteins found in nature are made from the same 20 building blocks called amino acids. And all amino acids have the same chemical structure, with the one exception of their side chain. So all amino acids have a central carbon with a hydrogen. They have an amino group, An amino group is a nitrogen with 3 hydrogens, amino group, and it has a carboxylic acid group, C double bond O with a negatively charged oxygen. So amino acids are called amino acids because they have a carbon with an amino group and then a carboxylic acid group. The difference, so all 20 look like that, but there's one difference. There are 20 different versions of a side chain, also called the R group. And each amino acid side chain has specific properties. This side chain could be negatively charged, it could be positively charged. It can be polar, like hydrophilic, or it could be non-polar, hydrophobic. And that really determines how these amino acids can interact with each other in a protein. And that determines the protein's structure. And we know the protein structure will determine how it can function. Right? The interactions between many amino acids determine the protein shape and function. So here's a list of some amino acids. You can see the 3-letter abbreviation and a 1-letter abbreviation with all 20 amino acids, and they're categorized by negative, positive, polar, or non-polar. And all amino acids in a protein are connected by peptide bonds. So when amino acids join together by peptide bonds, we call that a polypeptide. And when a polypeptide folds into its proper shape, we call it a protein. And like I said, structure determines function. So the protein's structure will determine how it can function. So for example, hemoglobin has a very complex structure, and that structure allows it to transport oxygen. Right? If it didn't have the structure, it would not be able to grab onto oxygen in the perfect way. In fact, red blood cells are shaped this way because of hemoglobin structure. If you have one little change in the structure of hemoglobin, suppose I just changed the 6th amino acid in hemoglobin from glutamic acid to a valine, that will cause the entire hemoglobin protein to fold incorrectly. If hemoglobin folds incorrectly, that means the red blood cells will have an incorrect shape. If red blood cells can't deliver oxygen to your body properly, that will cause anemia, right? Oxygen deprivation. So sickle cell anemia affects the entire body, but as a result of just one amino acid being switched in one protein out of tens of thousands in your body. So protein structure is very important. So enzymes are special types of proteins that are required for cells to speed up biological reactions. So for example, enzymes are actually going to do the dehydration synthesis and the hydrolysis reactions. They're going to help add the water to break the bond or remove the water. So enzymes speed up reactions. So for example, the detergent in your dishwashing soap has enzyme to speed up the breakdown of the fats and proteins in your dirty dishes. An enzyme speeds up a chemical reaction by binding to the 3D substrate. The substrate is the starting material at its active site. So every enzyme has a special place called the active site that can bind a substrate, the starting material. And what it does is the enzyme lowers the reaction's activation energy. So that's a little complicated. Again, it's a review of past biology lessons you've had before, but every reaction has an activation energy that's pretty high. But what an enzyme does is it makes it more likely for the reaction to proceed by lowering the activation energy. And enzymes themselves are catalysts, meaning they speed up the reaction without being consumed or changed themselves. So for example, suppose you have substrates, they bind to the active site, which is the 3D portion of the enzyme that can accept the substrate. And the enzyme will physically change its shape to facilitate a chemical reaction. So it will make the reaction happen a lot faster than it would have on its own. Right, and we said that all reactions have to overcome a barrier called the activation energy, and enzymes speed up reactions by lowering the activation energy. The way that enzymes lower activation energy is by physically they're physically moving, they're changing their conformation to make the substrates more likely to react, right? Enzymes facilitate the making and breaking of bonds. So for example, if my reaction were just getting frogs from one side of this barrier to the other, right? Suppose I have a really high barrier of getting the frogs from one end to the next. Right, it'll take a really long time for the frogs to jump over one by one. With an enzyme, it just lowers the barrier. It's the same thing, it'll just make it a lot faster. So that's the same concept. You might have a very high energy, in order to break down this you're breaking down this reactant into its separate products. It might take a lot of energy without an enzyme. But with an enzyme, it makes it a lot faster, a lot more likely for it to occur. So we said an enzyme binds to the substrate at its active site. So let's use an example of sucrose being broken down into glucose and fructose. So sucrose is the disaccharide and our body needs to digest it, so we need to break this bond. And the enzyme that breaks down sucrose is called sucrase. ASE ending is an enzyme. So a sucrase enzyme has a place that perfectly will bind to the sugar sucrose. So the substrate, the sugar, will bind to the enzyme, and we know that to break down sucrose, we need to do hydrolysis. But it's pretty hard for a water molecule to find this exact little bond to break. So what happens is the sucrase enzyme facilitates the hydrolysis reaction by almost positioning the water molecule exactly where it needs to be for it to break the bond. Enzymes do not have hands, but again, the enzyme can physically change its conformation, it could change its shape to make the water molecule able to break this bond and thus digest or catabolize sucrose into its two building blocks, glucose and fructose. The last category of biomolecules are nucleic acids. And nucleic acids are for storing, replicating, and processing genetic information. We need genetic information in order to make proteins. Right? We said proteins do the work of the cell. They can have structural functions or they could be enzymes. But the instructions for how to make proteins are contained in nucleic acids. And there are two major flavors of nucleic acids. We have deoxyribonucleic acid, or DNA, and we have ribonucleic acid, or RNA. And inside of our cells, we have a nucleus that contains chromosomes. Those chromosomes, if you were to unwrap them, are made up of DNA. DNA is deoxyribonucleic acid. And nucleic acids are polymers of nucleotides. So a nucleotide is a monomer of nucleic acids. And each nucleotide monomer has three components to it. There is a sugar. There's a deoxyribose sugar in the case of DNA, or a ribose sugar in RNA, but there's always a sugar. There's a phosphate group. That's a phosphorus atom with oxygens. And there is a base, not to be confused with like an acid and a base, but this is a base. There's one of four different bases we have in DNA and RNA. So in DNA, we have four bases that are thymine for T, cytosine is C, adenine is A, and guanine is G. So in addition to a sugar and a phosphate, each DNA nucleotide will have one of these four bases. Nucleotides arrange themselves in strands. So this is one DNA strand, and you can see each nucleotide attaches to the next by aligning their sugars and their phosphates. So here's one nucleotide, and then the sugar and phosphates stack up like the rungs of a ladder, the edges of the ladder. So the nucleotides are joined by their sugars and phosphates as a backbone. We call this whole thing the backbone. And then just to show you the bases, the nitrogenous bases, the bases have nitrogen, so they're called nitrogenous bases, kind of stick out like the steps in the ladder. And DNA is found as a double helix in its most stable form, which is two separate DNA strands that are weakly drawn to each other. Well, they have weak hydrogen bonds attaching the adjacent bases, and that keeps the two strands together. So hydrogen bonds are alone weak, but together, this keeps the DNA strand very highly intact. So a double helix is two strands of DNA held together by hydrogen bonds between the bases. Right, and like I said, genes are made up of DNA, and those A's, T's, C's, and G's make up the language or the alphabet of genetic instructions. So genes are made up of these four letters, A, T, C, and G. The order of these letters can determine the sequence of amino acids in a protein. RNA is a little different than DNA, like I mentioned, because it has a different sugar. It has a ribose sugar that has an extra OH instead of DNA. It has deoxyribose, does not have an oxygen here. Also, there's no such thing as thymine in RNA. It has a uracil, a U instead of thymine. ATP is an energy molecule that you've heard about before. It's the energy currency of the cell. ATP stands for adenosine triphosphate. It's a special nucleotide because it has a sugar. It's a ribose sugar. It has a base. It's an adenine base, an A. But instead of one phosphate group, like a normal nucleotide, it has three phosphate groups. And the energy is stored in the chemical bonds of ATP, specifically this last phosphate bond. This is a very high energy bond. So when you break off this phosphate, a ton of energy is released that can be used by the cell. So breaking the last phosphate bond can release energy the cell can use. And then we'll get ADP, adenosine diphosphate, with just two phosphates. If I then add on another phosphate, I can then remake ATP. So to summarize, we said carbohydrates include polysaccharides, disaccharides, and monosaccharides. Lipids include triglycerides to make up fats. Proteins are made up of amino acids. Nucleic acids like DNA and RNA are made up of nucleotides. And ATP is a special high energy compound that's like a modified nucleotide. So that is the end of chapter 2. Please review the textbook because it goes in a lot more detail that you may know, but use the learning map as a guide to make sure you understand the basics of the chemistry of life.